Welcome to another video on biology by Perfect Scores. This is Preetinder Kaur, and currently the videos that we are doing are based on the activities that happen inside a cell and the various biochemicals that are used up in our cells. So in this video we are going to discuss the concept of respiration that takes place inside cells. So we are going to focus on cell respiration. Now, if you define cell respiration, it is nothing but the controlled release of energy. So let me write that down, controlled release of energy, and that is from the energy compounds. That means from the organic compounds. And this energy that is released from the organic compounds is released to form ATP, which is adenosine triphosphate. So energy is released in the form of ATP. Now in cell respiration, glucose in the cytoplasm is broken down by a method known as glycolysis into pyruvate and with production of ATP, which is the energy released. So basically we break down one molecule of glucose, which is a 6-carbon sugar, into two molecules of pyruvate, which is a 3-carbon sugar, and a net yield is of 2 ATP. That's because 2 ATP are used up here and 4 ATP are released, so the net gain is of 2 ATP. This also results in the reduction of two hydrogen acceptors, that means NAD positive, into two molecules of NADH plus the hydrogen ion. So let's go ahead and discuss the various processes that are involved during cell respiration. And during anaerobic cell respiration, there's a different process that is followed. Anaerobic means in the absence of oxygen. In this case, the glucose is converted into pyruvate, and pyruvate can be converted into lactate or ethanol and carbon dioxide, and there is no yield of ATP. This happens during when there is an absence of a ready supply of oxygen, for example during intense physical activity when the reserves of oxygen are depleted. Now to generate the small amounts of energy provided by glycolysis, the pyruvate it must be converted into another substance before more of the glucose can be used. And this is because the conversion of pyruvate, so the pyruvate conversion, it replenishes the levels of the NADH that are needed for glycolysis to occur. Since it might seem confusing right now, so I'm going to draw a diagram to compare the anaerobic respiration between plants and animals. So that is what we are going to do now. Anaerobic respiration between animals and what happens in plants. So this is what happens in case of animals. The glucose through the process of glycolysis is converted into a pyruvate, and in this process two ATP molecules are used up to form two ATP molecules. So this is the energy, and then the pyruvate in the absence of oxygen gets converted into lactic acid, and two NADH are used up, and the cycle goes like this. So this is what happens in case of animals. Now coming over to plants, the glucose gets converted into pyruvate. Again, two ADP are used up to form 2 ATP. So that is the net gain. And pyruvate, in the absence of oxygen, gets converted into ethanol and carbon dioxide. And again, 2NAD, they are used up to form two molecules of 2NADH. So this is the process through which you can explain anaerobic respiration. Now the conversion, it occurs in the cytoplasm. The conversion of pyruvate, it occurs in the cytoplasm of the cell, and in animal cells, lactate, which is a 3-carbon compound, is produced. In plants, ethanol, which is a 2-carbon compound, and carbon dioxide is produced. And the conversion of pyruvate into ethanol and carbon dioxide, it is also known as fermentation. Now coming over to what happens during aerobic respiration, we are going to do that in detail. Now aerobic respiration occurs in the presence of oxygen and it takes place in the mitochondria. So it starts with glucose, which is converted into pyruvate through the process of glycolysis, which occurs in the cytoplasm. This pyruvate, it is produced along with 2 ATP molecules. When aerobic respiration takes place, that takes place in the mitochondria, and the result of aerobic respiration is carbon dioxide and water, along with 36 to 38 molecules of ATP being produced. And when it is anaerobic, that is something that we've already done, that takes place in the cytoplasm. And there are two results. In animals, it gives lactic acid, and in plants, it gives rise to ethanol and carbon dioxide. So that sums up the entire process of respiration. Now coming over to the next process, that means photosynthesis. It's a very important process, and the basic equation of photosynthesis is that 6 molecules of carbon dioxide and 12 molecules of water are used in the presence of sunlight and chlorophyll to make 1 molecule of glucose, 6 molecules of oxygen, and release 6 molecules of water. It's a 2-step process. The step number 1, light dependent reactions. Now these reactions, they convert the light energy into chemical energy, that is ATP. And then step number 2 is known as a light independent reaction, because it can take place in darkness as well. They use the chemical energy of ATP to synthesize organic compounds, that is glucose. So we are going to discuss these in detail, and the organic molecules that are produced, they can be used in cellular respiration to provide energy, which we have just done. The light that the plants get, it is derived from the sun, and it's composed of a range of wavelengths. So the visible spectrum of the sunlight, suppose this is the visible spectrum, anything apart from the visible spectrum that has wavelength of more than 700 nanometers becomes IR, that means infrared, and anything that has a wavelength of less than 400 nanometers becomes UV, which is ultraviolet. So wavelength goes like this and it starts contracting like this. So this is the visible spectrum that you can see in the middle. Now sunlight is white light. It's made up of all the colors of the visible spectrum, and each color has a different wavelength of light, and the wavelength ranges from 400 to 700 nanometers. So the colors would be violet, indigo, blue, green, yellow, orange, and red. Coming to the pigments that are present in plants to support photosynthesis, the pigment that is important in plants is chlorophyll. The chlorophyll is the main site of light absorption in the light-dependent stage of photosynthesis. There are a number of different chlorophyll molecules, each with their own distinct absorption spectrum. And when chlorophyll absorbs light energy, it releases electrons. And these electrons are used to make ATP, which is nothing but chemical energy. The main colors that are absorbed by chlorophyll are red and blue. The main color that is not absorbed, in fact it is reflected, is green. That is why plants appear green to us, and that explains why the leaves are green. The exception is when there is presence of other pigmented substances that produce a different color, for example red colored leaves or yellow colored leaves, and those extra pigments are known as anthocyanins. When these pigments are present, the leaves may have another color apart from green. The one thing to know here is that deciduous trees, they produce high amounts of chlorophyll in the winter as well because sunlight is insufficient. That allows the other photosynthetic pigments, for example xanthophylls and carotenoids, to come in the front, and that changes the color of the leaf in the winter. So these are two extra pigments apart from chlorophyll that are present. And now what we are going to do is a rough idea of how light is useful in photosynthesis. So light basically acts on the water. That is called photolysis because it gives rise to 6 oxygen atoms, molecules, and hydrogen ions. And these hydrogen ions are used for NADPH. And light also helps to excite the chlorophyll, which releases the electrons that are needed in ATP. And electrons also travel from water after its photolysis to the chlorophyll. Now, the first part of photosynthesis is the light-dependent reaction, which uses the light energy to make ATP. So what are the steps that are involved in the light-dependent reaction? The first thing is that light, it stimulates chlorophyll to release electrons, which results in production of ATP. Light also splits water by a process known as photolysis into oxygen and hydrogen ions. The hydrogen, it is taken up by a hydrogen carrier which is NADP positive to form NADPH. The splitting of water also releases electrons which help to replace those that were lost by chlorophyll. So whatever electrons were lost by chlorophyll, they are replaced by electrons coming from the photolysis of water. Now this ATP and the NADPH, they are taken to the site of the light independent reactions. So this is what happens in the first step, which is the light dependent reaction. The products are ATP and NADPH, and they go to the light dependent reaction. So this is the second part, and this reaction makes organic compounds from the products of the previous light dependent reaction. So carbon molecules, they use the NADPH and they use the ATP and they use the carbon dioxide from the atmosphere and they give rise to organic compounds. So the processes that take place are that first of all, the ATP and the hydrogen that was carried by NADPH, they are used to fix carbon molecules together. So fixing of the carbon happens at this place, and carbon dioxide is added to the basic carbon compounds, and this helps to produce more complex organic molecules, that is sugars, and they can then be stored for use in cellular respiration as and when required. Now the rate of photosynthesis, it can be measured by changes in the amounts of inputs, for example carbon dioxide, or the outputs, for example oxygen or glucose, of this photosynthesis equation. Now water cannot be measured because it's involved in a number of other processes besides photosynthesis, for example condensation or hydrolysis. So basically the input of carbon dioxide or the output of oxygen or the output of glucose can help us find out what is the rate of photosynthesis. Now, how to measure the uptake of carbon dioxide? It can be measured by placing a plant in an enclosed space with water. So place a plant in an enclosed space with water. Carbon dioxide is going to interact with the water molecules and give rise to lots of bicarbonate and hydrogen ions. They increase the acidity of the situation of the solution. Change in the pH value of that water can give rise to the measure of carbon dioxide that has been taken. So increased carbon dioxide uptake, that means the acidity will reduce, that means more alkaline pH, that means more than 7. How to measure the production of oxygen? Now oxygen production can be used by submerging a plant. So you submerge a plant in an enclosed space with water, and it should be attached to a sealed gas syringe. If oxygen gas is produced, it will bubble out of the solution and it can be measured by a change in the water level. Now these are two direct methods of observing the rate of photosynthesis. There is also an indirect method which is measuring biomass. So in this case, the glucose production can be measured by change in the plant's biomass or change in the plant's weight. This requires the plant to be completely dehydrated before weighing. So you need to dehydrate the plant completely before you weigh it to ensure that the change in biomass, it shows the change in organic matter and not the change in the water content. An alternative method for measuring the glucose production is to determine the change in starch levels of a plant. So this is another indirect method to measure the change in starch levels, because glucose is stored as starch. This can be identified via staining with iodine, because the resultant solution, it turns violet and purple, and then it can be quantified using a colorimeter. And coming to effect of other things on the rate of photosynthesis: temperature, light intensity, and carbon dioxide. The photosynthesis is controlled by enzymes that are sensitive to temperature. As temperature increases, the rate of photosynthesis will also increase. But above a certain temperature, it will decrease as essential enzymes, they begin to denature. So if you draw a graph, it's going to increase for a while and then come down, because at this point, denaturation of enzymes takes place. So this is the rate of photosynthesis and this is the temperature. This is the second factor, light intensity. As light intensity increases, the rate of photosynthesis will also increase till a certain point when photosynthesis is proceeding at the maximum rate. So if you plot a graph, rate of photosynthesis and light intensity, the graph is again going to be like this. Further increase will not have any effect on photosynthesis, so the rate is going to remain like this and not like this. It will not fall down, but it will not have any effect on photosynthesis. The reason is that the chlorophyll is already saturated by light. It is working with the maximum capacity, and extra light is not going to increase the rate of photosynthesis. Now, different wavelengths of light have different effects on the rate of photosynthesis. For instance, green light is not at all used, and that is the reason why plants reflect back the green light. And the third important factor is concentration of carbon dioxide. Now, as concentration of carbon dioxide increases, the rate of photosynthesis also increases till a certain point. After that, if carbon dioxide increases, it will not have any effect of the photosynthesis because the enzymes, they become saturated. So these would be the three resulting graphs that show you the dependency of the rate of photosynthesis on these three factors. So I hope this was clear how these different factors affect the rate of photosynthesis. And that is all that we'll be covering in this video. So we've discussed about cell respiration, aerobic and anaerobic both, and also the processes involved in photosynthesis. We might do them in detail in another video, but this would be a basic idea of how the processes take place. So thank you so much for watching the video. Don't forget to visit us at perfect-scores.com, to share and like us at Facebook, and to send any kind of suggestions or feedback at this address. Thank you so much for watching this video again.